and I'm going to be talking about genome-wide profiling of translation initiation and protein synthesis. So, of course, gene expression ultimately is the production of functional proteins in the cell from the information encoded in the genome. But when we measure gene expression, especially when we measure gene expression globally, we're actually usually looking at what mRNAs are present in the cell, not actually what proteins are being made. And that's largely because we have many good techniques to look at what mRNAs are around. Of course, microarrays and more recently, RNA-seq. But there's a lot of important information about what proteins the cell is actually making that we can't tell just by looking at what RNAs are present. And there are some recent global analyses that really highlight this. For instance, the Cellbach lab did matched RNA-seq and global mass spectrometry on cultured mouse cells, and they were able to look at the number of mRNAs and the number of protein copies of different proteins, and there's a real but far from perfect correlation between the number of mRNAs that are present in the cell and the actual amount of protein. And in this experiment, they were actually able to use pulse labeling to look at this in a little more detail. The idea is that you can pulse with either a nucleotide label or an amino acid label in order to monitor separately RNA synthesis and turnover, and similarly for protein, and really partition how these different factors contribute to setting the steady state level of protein in the cell. And what they found is that actually sort of gene-to-gene differences in translation played at least as large a role as gene-to-gene differences in transcription and mRNA abundance in determining the amount of proteins in a cell. And they sort of quantify that variation here. And of course, it's always going to be true that if you don't transcribe an mRNA, that you aren't going to make that protein. But there's quite a bit of room here for setting the sort of the quantitative amount of different proteins that you have in the cell by controlling how well the RNAs are translated. And in order to look at that in higher resolution, developed the ribosome profiling technique while I was a postdoc in Jonathan Weissman's lab. And the idea here is to isolate mRNAs with the ribosomes that are actually translating them in the cell, still bound in the positions that they have in the cell, and then to treat those with nucleases that are going to chew up most of that RNA. But about 30 nucleotides of that RNA is actually physically enclosed by the ribosome and is protected from nuclease digestion. And so after treating with RNases, we can purify out these ribosomes and then recover the RNA footprints of those ribosomes and analyze them by deep sequencing. And in one experiment, we can get, at this point, 100 million of these sequences from these ribosome footprints. And we can take those sequences and map them back to the genome, and we can get a couple of different kinds of information. So, of course, each ribosome in the cell produces one footprint, and then we count those footprints by sequencing, and we can simply ask, first of all, what is being translated? What portions of the complicated mammalian transcriptome are actually being physically occupied by ribosomes? And we can also ask how much. By counting the number of footprints that we get on different genes, we can ask how much different proteins are being made and how many ribosomes are committed to synthesizing that protein. And so today I'm going to talk about using the ribosome profiling of in vivo translation in order to measure gene expression. I think that's really going to be the broadest application of ribosome profiling, and in almost any experiment you can think of where you might want to ask about cellular physiology using gene expression profiling. I think ribosome profiling would be applicable. We can also use it to annotate what sequences are being translated, and finally, we can gain some mechanistic insights into the process of translation in vivo. So to start out, to give you sort of a sense of the experiment, so when we isolate these ribosomes and look for the footprints, one thing we actually see is that there's quite a bit of rRNA present here. So this is just a gel of from one of these experiments, and the ribosome footprints are actually very hard just looking at this to distinguish where they are from all of the sort of background RNA. we can look at the sequences that actually map to protein coding genes in order to look at the length of these ribosome footprints. And we see that they typically have a very narrow size distribution, that they're 28 to 29 nucleotides long in a pretty consistent way. And we can then go further and actually take these sequences I mentioned, that we can map them to the protein coding genes in the genome, and actually now take each footprint and ask where it falls, relative to the start codon of the gene it's aligning to. And so essentially this is the equivalent of taking all the genes in the genome, lining them up at their start codon instead of averaging how many footprints you get. And what we see is that there are a great many genes whose footprint, there are a great many footprints whose 5' ends, we're looking here specifically at the 5' end of these footprints. I'm going to, turn this off. Sorry. So the 5' end of these footprints typically show up starting 12 to 13 nucleotides upstream of the start codon. And we know that when the ribosome is initiating translation, so there are basically two sort of active decoding sites in the ribosome. This is the P site where the peptidyl tRNA is bound, and there's the A site where a new aminoacyl tRNA is coming in. So this is essentially the codon. The A site is the codon. It's being decoded by the ribosome at a given time, but at initiation, the start codon actually starts in the P site of the ribosome. And so we think that essentially the configuration of the ribosome at initiation, looking something like this, there are about 12 nucleotides upstream of the start codon that are protected, and then we have the P site and the A site essentially right in the middle of the footprint, and then some additional RNA on the 3' end protected as well. And so that sort of assignment, that calibration of where the decoding center of the ribosome is relative to the footprint, we can actually follow throughout the gene. And what we can see, you can sort of see starting right here, is that there's actually a 3 nucleotide periodicity to where the ribosome footprints show up. And we can quantify that if we're looking specifically at the 28 nucleotide footprints. So these are the ones that we think have been really sort of trimmed flush at both ends of the ribosome. We can see that the 5' ends of those have a very strong tendency to start on the first rather than the second or third nucleotide of a codon, meaning that we can really actually see as the ribosome takes a 3 nucleotide step down the transcript. And finally, at the stop codon.
So the remaining 25% is sequencing inaccuracies?
It's not sequencing inaccuracies. I think it's digestion inaccuracies. So what happens is that you can imagine that the footprint is it's 28 nucleotides, but it's sort of plus or minus 1 on each end. And so what you can have is, of course, they're 28 to 29 nucleotides, which means that sometimes they're extended, but they can also be slightly shorter on one end. And so sometimes if you're longer on the 3' end but shorter on the 5' end, then it looks like a 28-mer, but the whole thing is shifted over by 1. And similarly on the other end, and so that's what we think is going on here.
Do you do a size selection for this, or do you just sequence with
we do a size selection. It's a lot broader than this range, so we typically take between about 25 and 35 nucleotides. You can see that this, first of all, if we didn't do a size selection, we got a huge amount of junk from the ribosome. The ribosome, of course, has about 5,000 bases of structural RNA in it, and the footprint is just 30, so you'd be sequencing almost all our RNA if you didn't do something to make it more specific. And by cutting out just this, actually these are essentially our markers here to cut between 25 and 35 bases. But when we do that, even though we're sort of cutting in this region, you can see that the footprints specifically have a much tighter size distribution, even though if you look at, say, the RNA contamination, it really spans the whole range that we're selecting. And then we can actually also look in a similar way at the stop codon. And again, the translation terminates when the stop codon itself is in the A site. There's no tRNA that can recognize a stop codon. Instead, it's actually recognized by a protein release factor that docks into the ribosome in the same sort of way. And so termination happens when that decoding A site is sitting on the stop codon, and our sort of assignment of where the A site is in the footprint exactly corresponds to this very large accumulation of ribosomes that we see in this experiment. So, based on this, we think we're really getting single codon resolution about where these ribosomes are, as well as a sense of which reading frame they're decoding, at least in a statistical sense. If we have enough footprints, we can tell that. And it gives us this, and this is just from a different data set in mammalian cells, just sort of an overall picture of the density of ribosomes as they initiate translation on the stop codon, take 3 nucleotide steps down the message, and then terminate at the stop codon. So, the fact that we can see this, very clear enrichment in coding sequences in a 3 nucleotide periodicity convinces us that we're really following, translating ribosomes in vivo. And so far, I've been showing you just sort of whole genome averages, but we also want to look at specific genes, and the first question I'm going to ask is to essentially count, the number of footprints we, yeah.
Do you think the enrichment of the translation substudy is because there's much more pausing on the ribosome on this
Let's
side?
start. Yeah, it's a good question. I imagine that both initiation and termination are slower than elongation typically. The tricky thing, though, is that we see changes in the relative height of the start and the stop peak depending on really sort of details about how we prepare the cells. And as a result of that, we don't like to sort of really interpret those things strongly about what's going on inside cells. So, you can treat cells with elongation inhibitors, and when you do that, you see a bigger accumulation of ribosomes in the start codon. Probably what's happening is that initiation can continue. And even a very brief treatment with elongation inhibitors causes all the ribosomes to stack up on the start codon. And if you don't treat them with elongation inhibitors, it's more like this situation that I'm showing you here, where there's a substantial but smaller accumulation at the start codon, like this, and then a very large accumulation at the stop codon. And I don't think we have a great way of telling which of those is more physiological. Yeah.
You may have said this at the beginning, but are you cross-linking the RNA in the ribosome?
Oh, we don't cross-link at all, actually. The elongating ribosome is actually very stable, so we don't need to cross-link. We sometimes treat with elongation inhibitors like cyclohexamide or emetine, and that's, to some extent a technical thing to try to capture something like in vivo ribosome positions. So as I mentioned, those can have artifacts, but so the ribosome bound to the RNA is stable at room temperature for hours. And I guess one way I think, the ribosome is extremely processive. It can successfully synthesize, 10 or 20,000 codon genes, and so the sort of translating ribosome really stays stuck to the RNA well.
So it's still translating during the course of your experiment?
It's not translating during our experiment for a few reasons. We have no energy source, and there are several GDP-dependent steps involved in translation. We don't have any charged tRNAs present, and the whole prep gets diluted about 5-fold when we lyse the cells. So we're pretty sure there's not active elongation once we lyse the cells, but the ribosome is still in a translating conformation. In fact, people can do experiments where they isolate these, and instead of just putting them in some sort of generic molecular biology buffer, you can actually put them back into a system with an energy source and with tRNAs, and they'll continue to translate. They'll run off and finish synthesizing the proteins they're making. So they're competent to elongate, but we don't give them all the stuff they need to.
Are there any genes for which you don't see repetiticity like this?
There,
Imagine that you have multiple frames. Of cell experiments.
Yeah, we've done some stuff to look at that. And so there are a few pretty famous cases of this. There are some genes with programmed frame shifts in them. And those are sort of most prominent in some viruses. But there are also cellular genes where they occur, and we can see those. There are also some, for instance, the P16 ARF protein has actually, based on two different alternate promoter usage, it has two different transcripts that are read in two different reading frames. And we can see that signal, as well as the thing we see most when we look for this are cases where there's a strong out-of-frame upstream initiation site, and so you get translation and overlapping reading frames. But we think that the second product is probably not functional as a rule in cellular genes. It definitely does happen in viruses, though. So one of the limits really in any sort of deep sequencing analysis is how quantitatively and accurately you convert the pool of RNAs that you're looking at into a DNA library that you sequence. We know that once you have this DNA library and sequence it, it's actually quite good at counting what was there. But especially in the case of these ribosome footprints, which are which are short, and because they're short, you can't, for instance, random prime reverse transcription on them. There are some significant technical problems to sequencing them. And sort of to talk a bit about these, because in sort of thinking about, especially analyzing this sort of data in sort of higher resolution, I think it's important to remember the experimental considerations that are driving some of this variation. And so what we wanted to do as essentially a standard to ask how well our method of turning ribosome footprints into a sequencing library, ask how that worked, is actually just to take total cellular RNA and subject it to random chemical hydrolysis at high temperature. And we think under these conditions that it's basically, we get fragments that are totally random, and we can select size range that matches the size range of ribosome footprints and really take them through the same library generation procedure. And what we expect to see is that we get pretty even coverage across the whole transcript. Each of these, each piece of the RNA is equally likely to end up in a fragment that then gets turned into the sequencing library in pretty even coverage. But if there are some regions that are captured very efficiently and other regions that aren't captured at all, then we would see peaks and valleys in this coverage plot. And this could come up, if certain sequences were, say, complementary to some of the primers that we need to use, they might be particularly good at being sequenced. And if the enzymes we use have nucleotide preferences for their substrates, there might be certain RNAs that simply aren't captured very well. And we wanted to ask how well we could do this. And so we actually found that there's quite a bit to be gained by optimizing the protocol of just making the sequencing library once we add the footprints. And so what I'm showing you in blue is our initial protocol. And you can see exactly these sorts of peaks and valleys that I was talking about, that certain regions of this yeast gene end up strongly overrepresented in the library, whereas others are almost totally absent. And we optimized the protocol in a few ways to try to improve this. Just from sort of a technical perspective, the process of sequencing these ribosome footprints is actually quite a bit like the process of sequencing a microRNA, in the sense that they are these small anonymous pieces of RNA. And so we started with some of the procedures that people use for microRNA sequencing that involve just sequential ligation of linkers to the 5' and 3' ends, so you can then PCR amplify and then sequence the product. And we found the most important thing is that instead of trying to attach sort of two separate linkers to each end, that if we attach a single linker and then essentially circularize the whole product, that we got a much more efficient and much less biased library generation protocol. And so that was really, I think, the key thing, giving us this much more even coverage across this gene. And so it actually compares quite favorably in some sort of external comparisons to the, in terms of the variation, across a gene. They, in their paper, called our approach the 3' split adapter method. And so we think that this is minimizing our sequencing biases, and this is going to be important later when we're talking about things like asking whether the ribosome pauses at one specific spot during translation as opposed to another. If you're in the situation like we had in the initial protocol, you could see this huge accumulation of ribosome footprints at this one spot and virtually none here, and you could think, maybe does the ribosome just spend a lot of time paused right there? And in fact, it could be that the ribosome spent equal time at each position in this gene, but your library generation method just capture these footprints much better. And so that's one of the reasons I think it's important to think about these biases when you're trying to look especially for more sort of fine-grained information. But the first thing that we wanted to do with footprinting data was actually to just average the number of footprints across the gene, just across the entire gene. And so here's showing some data from cultured human cells, and each point is one gene, and the x and the y axes are the raw count of footprints that we got in 2 biological replicates. And I think the main point here is that we can get extremely reproducible measurements of how many footprints we get in these sort of complete biological replicates. And so counting the number of footprints is going to be our basic measure of gene expression in this system. And to ask, what sort of errors that we get in this, we can take those genes that we think we have reasonably high expression. I think the cutoff used here was about this point, about 100 footprint reads, which in this case include, there are several thousand genes across that threshold, and we could just look at the ratio in the number of reads in 2 different biological replicates as sort of a comprehensive measure of all of the biological and technical sources of noise we get in this experiment. What we see is the typical gene shows only about a plus or minus a 20% difference in the biological replicate, and there's virtually nothing that's showing a twofold change up or down. What that means is that if we were to compare cells in 2 different conditions and see a twofold change, that we could pretty reliably infer that that was a real significant difference as opposed to a result of this is sort of the variation that we'd expect between biologically identical samples. So we can make these measurements very reproducibly, but the real question is whether we're capturing biologically important information by doing this. And of course, the motivation that I gave you for doing this experiment was to ask more accurately what proteins the cell is making. And in order to look at that, we turned to an external standard, mass spectrometry, to ask what proteins are present in the cell. And that's what I'm showing you here. On the y-axis are protein abundance measurements from Matthias Mann's group by mass spectrometry in yeast. And then on the x-axis are measures of mRNA abundance and of the amount of protein being made by ribosome profiling. And so what we can see is that just like in the data from the Selbach lab I showed you at the beginning, that there is a correlation between the mRNA abundance and protein abundance in the cell, but it's far from perfect. And importantly, we can capture additional information about protein levels in the cell by actually looking at translation. This correlation is substantially better. Now, of course, the correlation is still far from perfect, and there are a few reasons for this. First of all, there's certainly noise in our data set as well as noise in the mass spectrometry measurements. The other interesting thing, of course, is that this is measuring the steady state abundance of the protein, whereas here we're measuring the amount of protein being made at any point in time. So if you had a protein that was very unstable, you might expect that its steady state abundance was lower than you might predict just by the rate at which it's being produced. And in fact, we can see that some of these genes that deviate very strongly downward are proteins that are known to be unstable. And of course, as measurements of translation and mass spectrometry improve, this might even be an independent way of also triangulating degradation rates for proteins. So told you that the mRNA abundance and profiling data aren't, capture different kinds of information. And I think one important point is that if you want to just ask about gene expression, ribosome profiling alone tells you how much protein is being made. And so if you have a situation either where, starting from some initial condition, you have fewer mRNAs present in the cell, or you have fewer ribosomes per mRNA, but the same number of mRNAs present. In either of these cases, you'll get fewer ribosome footprints from this gene. So by comparing this number of ribosome footprints to this, you can see the change in expression. If you want to know whether that change is happening at the level of transcription and mRNA abundance, or if it's happening at the level of translation, you need to also do RNA-seq to look for these changes or the lack of change here. And so when we're thinking about translational control specifically, I often talk about this translation efficiency ratio, which is the amount of protein being made per mRNA. And so we can just look first at the translational efficiency so here, starting with the same data I was showing you before from budding yeast, we can compare the amount of protein being made to RNA abundance. And so what you can see is that there's a typical amount of protein made per RNA. This is just the median here, this line. And above that, there are a number of genes, especially some genes that have sort of very high mRNA abundance and also are very well translated. So efficient translation is up and inefficient translation is down on this graph. And so these represent things like components of the ribosome and glycolytic enzymes, other proteins that the cell really needs to make a lot of. And in order to make a lot of those proteins, the cell transcribes them very heavily and also translates those mRNAs very efficiently. And then there are a wide range of RNAs that have somewhat lower abundance and in some cases have a much lower rate of protein synthesis per RNA as well. And so here, in this sort of range here is the contribution of translation to the quantitative gene expression that you get. At the same mRNA level, you can have very different, by here almost 2 orders of magnitude differences in the amount of protein being made. Now, of course, there's another population of RNAs that show up down here. These are things where we actually, in this experiment, didn't detect any translation of those RNAs, even though they're in some cases present at reasonably high abundance. So we expect that many of those RNAs are going to be translated under other circumstances, and so this is almost a qualitative change in gene expression that can happen at the level of translation. I'll give some more specific examples of this later, but you can imagine that these are RNAs the cell keeps around to respond quickly to stress as it has the RNA present but isn't translating it, and under some sudden change in conditions, that RNA would be poised to be translated and help the cell respond very quickly.
Is this in yeast cells?
This is in yeast. The picture is similar, actually even a broader sort of range of translational efficiencies in mammalian cells. I probably have one of those graphs later here. And this is just looking at that translational efficiency number to sort of how it varies, something, that the whole range is something like 2 orders of magnitude in addition to this totally repressed population of genes, and that even in this sort of larger set of genes, there really are, large, tenfold or more differences in translation efficiency.
Can you tell from the sequence only, for example, whether genes translated or not, the signals that?
There are a few things that will predict that a gene will be poorly translated. I think one of the difficulties is just not having this sort of global data set. It has been hard in the past for people to really study this. I think that one thing that's certainly true is there are some genes that have upstream AUG codons that sort of interfere with translation of the downstream gene. I'll talk more about that later, but that's definitely something that can predict poorer translation. Genes with longer and more structured 5' UTRs, we also see some effect of that on translation. It's not a huge effect, actually, but it is certainly there. And to some extent, that might reflect a difference in what the structure of that 5' UTR actually is in the cell versus what you predict it is or what you might even measure it as experimentally in vitro. I think that RNAs are less structured in vivo. Yeah.
Well, I had a question. So when you talk about mRNA abundance,
Yeah.
did you say that you are taking into consideration mRNA degradation rates?
Yes, when we measure RNA abundance, we're really just measuring RNA abundance, counting the number of RNAs and thus including both the contribution of transcription as well as RNA decay in those measurements. Yeah.
When you do RNA-seq, do you only isolate cytosolic fraction, or do you do total?
We've done, typically we do total RNA. We've done some experiments to fractionate cytosolic RNA specifically and ask whether, does this RNA abundance versus protein synthesis, does it get better? When you look only at cytosolic RNA, it isn't a dramatic difference. I'd say there's a modest but pretty minor difference. And you imagine, obviously one thing that contributes to this discrepancy is there is a large nuclear pool that's not even subject to translation. That's one source of discrepancy here, but it doesn't seem to be the major one. I should say that was in mammalian cells. It's possible in yeast that's a bigger difference. Not sure what way I would expect that to go, whether I expect that to be more prominent in yeast or mammalian cells. So the next thing we wanted to look at, and here again, I'm going to start with some data from yeast, are actually changes in translational efficiency under different circumstances. And so what I'm showing here on the x-axis is the change in RNA abundance when we acutely starve yeast cells for amino acids. This is just like 15 minutes of amino acid withdrawal from cells that were previously growing in rich media. And then on the y-axis, looking at the change in translational efficiency. So this is the change in ribosome profiling data after we correct for all of these changes in RNA abundance. So here, translation change is going to be purely vertical, and transcriptional change is going to be purely horizontal. And you can see, first of all, that there are a number of genes here whose RNA abundance decreases and whose translation decreases further. So these are things that are being repressed at a number of different layers. And we look at what these large group of co-regulated transcripts are, we actually find that they're strongly enriched for genes that are involved in ribosome biogenesis, RNA processing, genes that whose products localize to the nucleolus, which is where ribosomes are made. And in fact, if we look at those, we see that it's actually a remarkably sort of concerted change in the expression of a number of these genes. So in a quickly growing yeast cell, one of the major things that it has to do is to make more ribosomes so it can continue to grow more quickly. And in fact, over half of its protein synthesis capacity is directed towards making new ribosomes. And when the cell suddenly enters starvation as limited amino acids to make new proteins, one of the most important things for it to do is to rapidly shut down the production of additional ribosomes. So these collection of genes are regulated, their transcript levels are regulated, their translation is decreased, and there's probably also post-translational control. It's all working together to slow down ribosome biogenesis quickly, and that's really one of the major components of this response. There are, a handful, a collection of genes whose translation seems to be strongly induced under these conditions. And looking at these genes, it's more heterogeneous. You can't actually identify very clearly using just Go analysis what these are, but if you look at specific genes, certainly their function makes sense in terms of the response to amino acid starvation. They're often things that are involved in either amino acid import or amino acid biosynthesis in the cell. And one notable gene here is GCN4, whose translation is, in this case, about 5-fold increased, even though its RNA abundance doesn't change. This is actually a very well-studied case of translational control in yeast, I'll talk about more later. And so GCN4 translation is very rapidly induced by amino acid starvation. It actually is upstream of many of the transcriptional changes here, especially many of these transcriptionally induced genes here. And so its translation is induced very early as a part of this overall gene expression response to starvation. We next wanted to look at changes in gene expression that might happen in mammalian cells. And one of the questions that we realized ribosome profiling was very well situated to answer was the impact of microRNAs on gene expression. And so this was in collaboration with Dave Bartel's lab. And the basic question we wanted to ask was that in different systems and under different conditions, people had reported that microRNA could either destabilize its target RNA or repress its translation. And it's further complicated by the fact that to some extent RNA stability and translational repression could be coupled. And it's actually very difficult to design experiments, especially in vivo, that can separate those two effects. But we wanted to see what we could see through ribosome profiling. And so here on the left, we're looking at the RNA-seq and ribosome profiling fold changes for 3 different sets of, really 4, but I'll focus on 3 sets of genes in this experiment. So in green are genes whose protein products are known to be decreased by this microRNA. And in blue are genes that have a predicted microRNA target site, though their repression hasn't been validated. And finally, in black are genes that don't even have a computationally predicted site for this microRNA. And so what you can see is that the genes with microRNA target sites, their RNA levels as measured by mRNA-seq, are typically decreased nearly 2-fold. Relative to the genes with no sites. And if we look at the ribosome profiling data, we see a similar decrease, and we can then look at the translation efficiency ratio for all of these genes, and we can see that there is a small but very significant decrease when you look in aggregate in the translation of these genes. So what that's saying is that RNA levels are going down, protein synthesis levels are going down a little bit more, and so the microRNAs under these conditions really are affecting both RNA stability and the translation of those RNAs, even after you account for the destabilization. And the approximate ratio is 5 to 1 under these conditions, that about, for any given gene, about 5/6 of the decrease in expression can be accounted for the decrease in RNA abundance, and there's an additional 6 that can be attributed to translational repression. And so these are all essentially looking in aggregate at all of the target RNAs. And we also wanted to ask more specifically whether it was really the same genes being affected, both the level of RNA stability and translation, or whether there were maybe subsets that were affected in one way but not the other. And so here, if you focus especially on the green points, you can see that really the change in translation, the change in RNA abundance, really sort of follow each other. So there isn't a class of genes, say here, whose RNA levels decrease but whose translation isn't affected, and there isn't a class of genes here whose translation is strongly repressed without a change in RNA abundance. And so I think that at least in general you can say that you can find the microRNA targets by looking at which, by looking at RNA level changes.
The position of the microRNA target within a transcript would not influence translation?
Doesn't seem to sort of cause it to influence translation and RNA stability differently. I think some of the more mechanistic work on this maybe suggests that it's really, in both cases, the interaction of the RISC complex with PAB that's responsible for both the destabilization and the repression. So it maybe makes sense that the sort of the magnitudes vary together because it's really mechanistically the same effect. But again, I think that's sort of harder to separate out.
I guess it's something special about year 155.
Yeah, we don't think so. They also did some other experiments with MIR1 and HeLa cells to try to ask whether it was specific to that microRNA. And so the argument is that RISC binding leads both to reduced translation, which accounts for about 1/6 of the repression, and also decreases the RNA abundance, which then accounts for about 5/6 of the repression. So actually, since this work was published, Antonio Heraldez has done some nice work showing that in a zebrafish developmental model, they could actually get essentially temporal separation of the translation and mRNA decay effect, so you can see the translational repression start earlier. They have a developmental system where a single microRNA switches on and is the only microRNA active at the onset of zygotic transcription. And by comparing wild-type fish with those that lack dicer and thus can't produce that first zygotic RNA, you can see the difference in the translation of those zygotic microRNA targets decreases first, and then their RNA abundance later decreases about an hour later. It's possible that this is happening in this system as well, that if we were to look at a very early time point, that we would see the reduced translation as a dominant effect, and that the decrease in mRNA abundance takes longer to set in. For technical reasons, we weren't able to look at sort of 1 hour time points here. These are all after 36 hours. But if we're talking about sort of the steady state effects of microRNAs, it does seem like they really are affecting both RNA abundance and translation. The ribosome profiling that I was doing first in mammalian cells was actually in mouse embryonic stem cells. And so I wanted to look at the translational changes that accompanied a more sort of physiological developmental process. And one of the things that you can do with embryonic stem cells is to differentiate them into embryoid bodies, which are a fairly heterogeneous collection of more differentiated cell types that emerge when you remove the growth factor LIF. And the experiment here was to produce these embryoid bodies and compare by ribosome profiling an early and a late time point in differentiation. And so here I'm showing you just the change in translational efficiencies that we see across the transcriptome at an early and a late time point relative to the ES cells we were looking at. So this early time point, this is actually a time when there are large changes in transcription. We don't actually see dramatic changes in translation, but if we look further out after 8 days of differentiation, we actually do see that there are substantial changes. There are many genes whose translation increases or decreases threefold or more. And if we look at this, there are a couple of things that stand out. One, as I mentioned, that there are broad range of changes in translational efficiency. The other is that it's sort of skewed, that there are many genes whose translation increases substantially, but only a small handful of genes whose translation decreases quite a lot. And it seems sort of paradoxical. The resolution to that paradox is to see that the genes whose translation is strongly decreased are, again, the ribosomal proteins, other genes involved in ribosome biosynthesis. So because these are very abundant, the sort of threefold translational repression of these ribosomal protein genes leaves a lot more biosynthetic capacity available to translate all the other genes, including the cell type specific transcripts that are being induced during differentiation. And again, it makes sense that ES cells are proliferating rapidly, and one of the main things they need to do is produce more ribosomes so they can keep growing, and that's no longer true in embryoid bodies, which are where cells are dividing much less rapidly. So did you?
Yeah, I was just asking you, it seems like most of the embryoid bodies, the lineage-specific markers are expressed much earlier than day 8.
Yeah, that's, yeah.
Do you think those are like especially
Regulated? They may be. So whether there's, well, I guess one problem is that the genes that are not, if they aren't transcribed at all in ES cells, we don't sort of have a point of comparison to ask how well they're translated in ES cells. You're right that they're definitely, it's, other people have certainly seen, and we could see in our data as well, that at this 36-hour time point, we do see a number of lineage-specific genes switching on. But I guess we see some of that. It's quite strong after 8 days. I'm not remembering offhand. We sort of pulled out several of the sort of marker genes. Just if nothing else, it's a validation that we were doing the experiment. We were getting the sort of differentiation we were hoping to see. I don't remember the details offhand, but certainly. I certainly have that data, and we can talk about it later. And so so the ribosomal proteins, again, show up as sort of a major set of target genes. And in ES cells, this regulation very likely is happening downstream of the kinase mTOR. So mTOR is a master regulator of cell growth and proliferation, and it integrates a lot of different environmental and intracellular signals, including ones probably quite important here, PIP3 signaling. and phosphorylates a number of different targets, including some involved in translation. And in this response, there's some pretty good evidence that 4-EBP is the key mTOR target here. So 4-EBP is essentially it's a repressor of translation. It binds to the initiation factor 4E, but when mTOR phosphorylates it, it can't bind 4E, and so translation is more active, and it's more active on these ribosomal protein genes in ES cells. And as embryonic bodies form, we know that mTOR signaling decreases, and this means that there's more active 4-EBP, and that 4-EBP represses translation and specifically drives the repression of these ribosomal proteins. So to some extent, the involvement of mTOR in these embryonic bodies is an inference based on what we know about ribosomal protein translation and about mTOR in ES cells. And we wanted to find a system where we could ask sort of more directly what mTOR is doing. And so we collaborated with Davide Rogera at UCSF to look at a cancer model where mTOR signaling is also quite active. mTOR is actually defined as the target of the drug rapamycin. But it turns out that rapamycin is actually not a very potent mTOR inhibitor. It's an allosteric inhibitor, and there's feedback control that actually sort of counteracts much of the effect of rapamycin, and so you get much better inhibition of mTOR if you have an actual active site ATP analog inhibitor, and Davide's lab was involved in characterizing one of these active site inhibitors and developing it actually to the point of being a preclinical cancer drug. And so they were very interested in looking at the translational effects of their drug and comparing them to rapamycin. And so we worked together, and here I'm just showing you that the changes in translational efficiency of all the genes in these cells following in the x-axis of the impact of rapamycin, so shift to the left are rapamycin inhibition of translation, and the effect of their active site inhibitor PP242, and here shifts downward are repression. You can see that these target genes, which include the ribosomal proteins, are modestly repressed by rapamycin treatment and strongly repressed by this active site mTOR inhibitor, confirming that this pathway really is on target to controlling these ribosomal proteins, and also in this cancer model a number of other genes that are involved in basically in cell growth and invasive behavior, that if you in these models of invasion that for instance the intermediate filament gene vimentin is repressed, and other genes, and so if you treat cells with this, they're less invasive. In addition, in fact, if you xenograft tumors into a mouse, they're also less invasive and less metastatic when you treat the mouse with this drug. This seems to be involved, so it seems to be a sort of a common pathway of translational control involved in ES cell growth and in tumor cell growth. And it's actually also something that's involved in sort of cellular activity, even in cells that are not dividing. So we looked at changes in translation in mouse liver, comparing the acute effects of glucose feeding, where we can see again the translational induction of the ribosomal proteins about 15 minutes after glucose feeding. And again, this is likely through the result of mTOR, as one of the signals upstream of mTOR is insulin signaling. So this is sort of a picture of one particular prominent translational control pathway. It's fairly easy to see, and in fact even to study without ribosome profiling, because it causes the concerted change in translation of over 100 fairly abundant mRNAs. And we were then interested in going on to characterize some other translational control pathways as well, things that are triggered in response to stress. It's nearly 5, so this might be a good point to
yeah, if you want, it's up to you.
Okay. I don't know, I think okay, actually I'll go through the next little bit then. Yeah.
We started with late, so
Okay. So
yeah, so
oh yeah.
for remember that there was some signals upstream of these MTOR
Yes.
targets, right? Is that
Yeah.
so to
Yeah, definitely. So what you
AU-rich signal?
it's actually pyrimidine rich. So the strongest targets are those that have basically 6 to 12 pyrimidines right as the 5' nucleotides of the transcript, this oligopyrimidine tract, which is a pretty distinctive feature of the ribosomal proteins. Some of the other targets, it wasn't necessarily right at the 5' end of the transcript, but near the 5' end of the transcript, there would be, again, this oligopyrimidine tract. It's not clear what protein is recognizing that. I think that would be, it's an excellent question. I'm almost certain there is some RNA binding protein that recognizes that motif. We don't know what it is at this point.
But it's not EIF4
No, the interesting thing about 4E, so 4E is the cytoplasmic cap binding protein. So it's believed to be present on every transcript in the cytoplasm. And it's not totally clear why 4E binding protein only affects these oligopyrimidine RNAs and doesn't affect every RNA. So whatever 4EBP is doing, it seems to be somewhat specific to those RNAs, even though 4E is not specific.
And the other question I have is about so some of these MTOR inhibitors are in clinical trials.
Yeah.
Pretty promising. Are you saying that ribosome is sort of shutting down ribosomal protein production is some high goal in this anti-cancer activity, or is this something else, some oncogenes or?
I think probably it is. I think there's a lot of feedback control that the cell can tell how much protein synthesis capacity it has. Actually, before people knew about cyclin-dependent kinases as control of the cell cycle, one of the major ways that people were studying how sort of cell division was gated in mammalian cells was looking at protein synthesis capacity. If you treat cells with low doses of inhibitors like cyclohexamide, the cells need to grow to a larger size before they'll divide and will divide more slowly. So there's some sort of feedback control there. And Davide, as well as Nahum Sonnenberg's lab, has some evidence for essentially a role of 4EBP as a tumor suppressor, and I believe phosphomimetic 4E as an oncogene. So basically, I think at least in mouse models, pretty directly implicating the 4E and 4EBP sort of circuit in transformation. MTOR affects many different processes. It probably isn't just translation, but I think there are a number of different lines of evidence suggesting that it is actually one effect of mTOR that's sort of on pathway to transformation. Another case that we were sort of known case of translational control that we were interested in looking at happens in response to unfolded proteins in the unfolded secretory proteins. And so the unfolded protein response, the stress response to essentially to damage to the ER, is biologically important in a number of contexts. It's, among other things, necessary for pancreatic beta cells to function properly, probably because they have a particularly large protein secretion burden. It's also important in B cells in the blood, which need to secrete very large amounts of antibodies. And really, it's important for sort of the normal health of most cells. And it's possible to trigger the unfolded protein response by treating with this drug tunicamycin, which blocks glycosylation in the ER. So one of the ways that proteins are matured in the ER is that sugars are attached to them, and some proteins fail to fold properly when they're not glycosylated. And so this drug tunicamycin, which is a natural product derived from some marine organism, poisons this pathway. And so when cells experience tunicamycin, experience the overexpression of a misfolded ER protein or a number of other insults, it triggers ER stress that activates the protein kinase PERK. So this is a transmembrane protein kinase, and its major characterized target is the translation initiation factor EIF2 EIF2E. And so phosphorylated EIF2E activates the translation of transcription factors, including ATF4. So ATF4 is actually the analog of the yeast GCN4 gene that I mentioned earlier. So it's this stress-responsive transcription factor whose translation is controlled in response to stress. And so ATF4 is known to induce the transcription of a number of genes. So it induces protein chaperones and other enzymes that help to fold proteins in the ER. It induces amino acid importers and other proteins involved in starvation response. And it specifically induces some other genes as well, including this transcription factor CHOP and this gene here, which is actually the protein phosphatase that dephosphorylates EIF2. So there's an interesting feedback loop here. Of course, phosphorylating this translation initiation factor actually broadly affects translation. So one of the things that happens here is the cell generally decreases the amount of translation that's happening, while specifically inducing the translation of a few target genes that are going to help it deal with this stress. And we wanted to more fully characterize this protein, which had a few known targets, as I mentioned, ATF4 and ATF5. And so again, we're showing ribosome profiling data comparing cells treated with tunicamycin to untreated cells. And on the x-axis are changes in RNA levels, and the y-axis changes in protein synthesis. And I'm actually, in this graph, plotting it slightly differently. So this is ribosome profiling data, not translation efficiency, so it's not corrected for changes at RNA levels. So transcriptional induction is essentially a shift both to the right, which is increased mRNA levels, as well as a shift upward as a new RNA is better translated. So you can see, and this was known before, is that one aspect of the unfolded protein response is the induction of immediate early genes, such as FOS and EGR, as well as I mentioned, the translational induction of ATF4 and ATF5, whose RNA abundance remains basically unchanged, but whose protein is synthesized much higher levels. So we wanted to look at what some of the other genes affected by this pathway are, of which there were several, not actually a huge number of genes, at least in these cells, which are MES for technical reasons I'll get to in a minute. And we actually found the other three genes that were specifically translationally induced, found a couple of interesting things. Firstly, so the transcription factor CHOP and this phosphatase that I mentioned before as late transcriptional targets of this pathway are also rapid translational targets. And so this suggests is actually, in addition to this long transcriptional circuit, that EIF2 alpha directly affects production of CHOP and of this phosphatase. It's going to feed back and decrease EIF2 alpha phosphorylation. And the last gene we found, SLC35A4, is a poorly characterized gene, but based on its localization and similarity to other genes, it is very likely to be an ER sugar transporter that transports UDP galactose into the ER. And this is actually the sugar donor that's going to be responsible for the ER glycosylation that tunicamycin is repressing. And so, at least under this particular stress, we can see the very specific translational induction of a gene whose molecular identity tells us exactly what tunicamycin is doing. Since there are other ways of inducing the unfolded protein response, we're actually right now doing some experiments to see whether this ER sugar transporter is also induced translationally under other conditions, or whether somehow the cell is distinguishing that glycosylation specifically as defective. But in any case, I thought that was quite interesting, and also that these feedback circuits are acting quickly into level of translation. We were working in these MEFs in part because we had access to MEFs from mice where the PERK kinase was knocked out. So these are essentially, very similar cells, but with no PERK, and also actually cells in which non-phosphorylatable allele of EIF2 alpha was knocked in. So instead of the phosphorylatable serine here, it has an alanine. And we can look at our data in both of these conditions, and we can see, first of all, is that loss of the ER kinase PERK basically totally abolishes the response. There are a few genes whose translation seems to be repressed, but basically nothing is either transcriptionally or translationally activated here. The other thing we see is that in the non-phosphorylated EIF2 alpha, we still see some of the transcriptional response. We don't see the translational induction of ATF4 or any genes like that. One interesting thing we see is that many of the transcriptional targets here, as I mentioned, their RNA levels increase and their protein synthesis levels increase as a new RNA is being translated. Here we don't see very good translation of the new RNA. So one possibility, I mentioned that EIF2 alpha is causing sort of a global decrease in translation. It might actually be sort of reserving protein biosynthetic capacity specifically for these newly induced RNAs. And when you don't have EIF2 alpha phosphorylation, even though you can upregulate synthesis of these RNAs, you can't translate those new proteins, and thus you can't as efficiently relieve ER stress. And this could be a more general feature of stress responses. You can imagine that in response to stress, cells need to rapidly produce RNAs that are going to help them deal with the specific stress, and they probably want to repress the translation of whatever sort of housekeeping or sort of standard genes that they were expressing before and really favor the translation of those. And that may be one role for EIF2 alpha phosphorylation, which shows up actually in a number of different stress responses.
Likewise, is there something special that these mRNAs
that make them? Yeah, so there is, and this actually relates to their, again, they have actually some of these upstream AUGs, and there's a sort of Baroque regulatory mechanism that controls their translation. So what happens basically, just to remind you that in eukaryotes, translation initiation happens when the ribosome loads on to the 5' end of a transcript and scans down that transcript to find the first AUG. And so as a result, if you have an upstream AUG, the ribosome will often translate that instead and never actually make it to the downstream protein. And so ATF4 actually has two upstream AUGs. One of them initiates translation of a very short, like 5 amino acid reading frame. Under normal conditions, the ribosome will actually translate this, and for reasons that aren't really well understood, after translating this upstream open reading frame, it's actually competent to resume scanning for a new AUG codon, and then it will encounter this second AUG codon starting the second longer decoy reading frame. It'll translate this decoy reading frame and never reach the actual ATF4 protein coding gene. Under conditions of ER stress, when there are when EIF2 alpha is phosphorylated and inhibited, the ribosome can sometimes bypass this second decoy AUG and instead actually make it to the main protein coding gene. And this is basically the standard model, which I think that's probably incomplete, but the standard model. So EIF2 alpha is a subunit of the complex that actually brings initiator tRNA to the ribosome. And when EIF2 alpha is phosphorylated, it's inhibited, and there isn't as much of this EIF2 initiator tRNA complex available. And the thought is that basically here, this ribosome isn't getting a new initiator tRNA in time to initiate here, and that's why it can sometimes bypass the decoy start codon and actually reach the start codon for the protein coding gene. So looking at these sort of upstream translation events is something that I think ribosome profiling is very good at, because most methods of looking at translation measure the number of ribosomes loaded onto an RNA, but don't give a great resolution of exactly where they are, whereas ribosome profiling, you can actually see exactly where that ribosome is and see how many are on this URF here and how many are on the protein coding gene, sort of ask some questions about this. So that is, I think, the next thing to talk about, but I think we should probably take a break. Yeah.
Questions?
Sorry,
Yeah.
what's the distance between the red open reading frame and the orange one?
Yeah, it is, I think this gene, it's 150 or 200 base pairs, something like that. And then this is, again, I think this distance is about 100. Actually may have on the next slide. Yeah, scale bar. Actually, so this is actually drawn to scale, and that's 100 nucleotides. So that's probably about 150 or so, and that's probably another 100 or maybe a little more. Yeah. Yeah.
I was curious to know, the previous slide, which RNA binding proteins are involved in regulation of where it starts, which start site units?
So the best characterized proteins, I mean, there aren't so much RNA binding proteins. What's well characterized are the 40th subunit is loaded onto the mRNA complex with a lot of initiation factors. There's EIF2 that I mentioned, EIF4 that came up earlier. There's an EIF3 and a 1 and a 1A and a 5, and all of these things are involved in sort of binding this complex, but ultimately what really recognizes the start codon is actually base pairing between the initiator tRNA and the mRNA. And so EIF2 is GTPase, and its GTP hydrolysis is actually coupled to the recognition of a start codon by the initiator tRNA. So it hydrolyzes GTP, and then some of these initiation factors are GTPase activating proteins for EIF2 or otherwise modulate EIF2's GTPase activity. And so that's really the key molecular event in start codon recognition. So to remind you where we left off, we were talking about the role of essentially decoy initiation sites as a way to regulate how much protein the cell is making by capturing ribosomes before they could actually get to the protein coding gene. And this is something that we can see pretty clearly in our data. And so here I'm just showing you actually these are profiles of ribosome occupancy across the ATF4 transcript in unstressed cells and in cells that have been poisoned with this drug tunicamycin that induces the translation of ATF4. And what you can see, first of all, is that there are a lot of ribosomes stacked up on this first upstream open reading frame. And you can also see a number of ribosomes on this second. Oh, yeah.
Are the scales identical?
Yeah, the scales are corrected between the two samples for things like read count and everything. So you should be able to make sort of y-axis comparisons between these two. And so what you can see, and first of all, if we look way downstream in the body of the gene, you can see there are substantially more ribosomes here than here. Still doesn't look like there are a lot of ribosomes there, but the difference is quite large, about 5 or 6-fold. And those correspond to the ribosomes that are actually making this ATF4 protein. But where we see more ribosomes are stacked up at essentially these decoy initiation sites. One, a lot of ribosomes stacked up here and quite a few here as well. There are a couple of interesting things. One is that the naive model I was describing to you before, you'd sort of expect that there would be fewer ribosomes here if there are going to be more on the gene, because it's essentially presented at least as a sort of a choice between the upstream open reading frame 2 and the actual protein coding gene. And that doesn't seem to exactly be what happens, and that there are actually probably more ribosomes on this upstream open reading frame here. So we think this is pointing to something about this model of regulation that doesn't fully explain what's going on. The other thing you can see is there's actually a site up here even further upstream than the first AKG of the transcript where we actually see some translation. And so this looks like it must be initiation somehow to non-AUG codon. And this is something that we actually see on many transcripts, and it's just, again, a reminder that initiation goes by 5' to 3' scanning. And when we do ribosome profiling, we see a number of these upstream initiation sites, and they request translation. And we actually saw this first in yeast, where there are cases like this gene where we could see an upstream open reading frame and there are ribosomes there as well as in the gene. We also saw cases where translation seemed to be initiating at a UUG codon on this transcript and causing the translation of some very short reading frame here. And we could actually take this and fuse it to luciferase, and we could see that when we put luciferase reporter in frame with that UUG, we made quite a lot of luciferase, and there's virtually none when we frame shifted it relative to that UUG. So in this particular case, this UUG codon can direct the production of a fair bit of protein. And so this actually, I mean, I'm showing you one example here that's actually affected many yeast genes, and then and one of these included the gene GCN4, which is actually, I'll just go through this quickly, but regulated in basically the same way as ATF4, that you initiate translation at the first UORF, and then normally the downstream UORFs suppress translation, but under conditions where EAF2 alpha is phosphorylated to reduce, it can skip past these upstream ORFs. And then we see this in ATF4 as well. And more generally, we see this happening actually on many genes. So the NANOG is the transcription factor that's involved in regulation of in pluripotency in the S cells. And you can see that there are actually quite a lot. So here the thicker line is actually the coding sequence. And here on the 5' UTRs, you can see that there are quite a few ribosomes there, even though there are no AUG codons in this 5' UTR. So there seems to be a fair bit of translation initiating, and like in yeast, we can fuse these 5' UTRs to a reporter like luciferase or GFP and actually see that they direct the synthesis of proteins. And this is one of the things that really came out of ribosome profiling that I think was slightly surprising to us, that there's a fair bit of translation in 5' UTRs, suggesting that UTR might not be the best name for them. And also that the initiation often seemed to happen at codons besides AUGs. And there are cases where human diseases are actually caused by mutations in UORFs that can affect the expression of the downstream gene. Actually, the loss of this upstream UORF causes higher expression of the HR transcription factor, which causes hereditary baldness syndrome. It's just sort of one example where it's very clear that it's the mutation in that AUG codon affecting translation that causes the disease.
Does that mean that there should be a longer proteins, or is it just premature sort of
well, so this in this
stable?
well, so this is like the ATF4 case, where this is sort of a decoy upstream start site. This orange shaded box is the short reading frame that's translated when you initiate here, and so what it's actually, again, it's just a question of in the normal case, many ribosomes translate this thing and so don't make any of this downstream protein at all. When you have the mutation, they more often skip this and actually express this transcription factor.
But in the nano case,
Yeah.
I mean, you did really see
Yeah.
translation all across
There's not some of it, yeah. But again, we think a lot of this is out of frame and doesn't. I mean, there definitely are cases where you actually make longer proteins, and a lot of this, though, is out of frame. And we think there's probably, in most cases, selection against actual N-terminal extensions in the protein when there are possible problems for it, yeah.
And this short peptides, I mean, they're not doing anything in this, is there evidence that
It's an excellent question. There's no evidence that they are, but it might be hard to find that. There certainly are short peptides that are functional, so it's certainly a possibility.
I guess you could scramble the sequence.
You could scramble the sequence. So in the case of ATF4, this, for instance, and in GCN4, the sequence of those upstream open reading frames is not important, it's just their presence. But many of these other cases, it could be that these peptides are doing something important, yeah.
Just a question, do you preserve the strand identity?
We do, yes, yeah. So these are definitely on the sense strand.
I wanted to ask something related to the short peptides.
Yeah.
If it's possible, like, to measure how long they stay around, if they are really produced or it's just binding, but it's kind of stalled.
So actually looking for the short peptides turns out to be very hard for technical reasons, like by mass spectrometry. I don't do a lot of mass spectrometry myself. I'm told that it is very, very hard to go after these. I think there's a very high background of this junk in the cell, breakdown products, and the proteasome and things are in basically the same size range. And as a result, it's hard to look for them that way. And that's sort of why we did some of these reporter experiments. We can't directly detect that they're making this protein, but you can say, okay, if I leave this whole short peptide and then right at the end of it I stick like luciferase, I can easily detect that I make that luciferase and we're inferring that that means that in the normal case, the ribosome is actually translating that short protein, even if it might be immediately degraded. So.
Are there classes of genes that show this anomaly?
I think
or is
the...
this, like, ubiquitous?
I wouldn't say it's ubiquitous. There are many genes that show it, though, and I think mostly it's genes where you need to make a lot of the protein tend not to have this. It's probably just if you want the most efficient possible translation, you don't put all this other, you have selection against all this other stuff in the 5' UTR. In cases like nanog, the cell isn't trying to make as much nanog as it possibly can. It's trying to regulate the production of nanog in some very specific way.
We haven't seen how peaks
These guys, like these. The peaks we think are places that ribosomes pause. I actually have a bit more about that later. Yeah, we do think that in at least some cases you can see that something like that is a spot where the ribosome is spending a long time for some reason.
Do you think there's a function to this, or is it just kind of random?
I mean, I don't know. There are a few cases where it has a function, and people have proposed more generally that if you have, especially in a complicated multi-domain protein, the protein is folding as it's being translated. Translation is relatively slow, and you could imagine that you actually want to essentially stall the ribosome to allow an N-terminal domain to fold before the C-terminal domain is even synthesized to avoid inappropriate interactions between them. And something that
those lines you see an enrichment in this internal stuff, pausing sites between domains, or did you guys look at that? So?
I think we tried to look at that. I think we had a hard time coming up with a large enough set of proteins whose domain organization was clearly known. I think it's a great question. We just don't know. Yeah.
Do you know whether those short peptides, do they come from the full length mRNA? Because there is this paper in yeast where they show those short mRNAs from those upstream feeding trains. Do you think it's kind of related?
I think it definitely could. Yeah, we can't tell what the transcript structure these things came from. So yeah, this is the paper from the Steinmetz lab showed that in some cases things that we would have called URs are actually part of actually independent short transcripts. So that's very possible too. I guess from our perspective, we can certainly say that these things are still peptides that are being made in the cell, but they might not have the regulatory effect we expect, either in that paper or in a paper that came out at the same time from Wendy Gilbert's lab. I think they had some pretty good evidence that the URs are repressive only when they're actually in the same transcript, and so the URs that were sort of independent entities as opposed to part of that transcript don't actually, of course, repress expression of the downstream gene. Yeah. So we wanted to look at translation initiation specifically. So when you're looking at one of these profiles, like in nanog, you could sort of look at this and say that, yeah, translation is probably starting here and here, something like that, and maybe see what codons are there. But we wanted a way to actually look at that a bit more cleanly. And what we did is use this drug herringtonin, which is going to freeze ribosomes right at sites of initiation. So the way it works is that it poisons peptide bond formation in the ribosome. Now, peptide bond formation happens at every cycle of elongation during translation, but it turns out that herringtonin, which is a pretty big molecule, can only actually get into the ribosome and into the peptidyl transfer center when the ribosome is disassembled before it initiates translation. So when you add the drug, all of these assembled elongating ribosomes are immune, and so a few minutes later, the poisoned ribosomes have been locked in place right on the start codons, and all the other ribosomes have run off. And if you do a footprinting experiment under these conditions, all the footprints you get will be showing up right after, just after sites of initiation. So we can do this, and so if we treat with this drug for, in this case, like 2.5 minutes, we see a massive accumulation of ribosomes right at the start codons that trails off over the first 10 or 20 codons. And we can then look at individual genes and see basically the same pattern. So after herringtonin treatment on this gene ACTR2, which is picked mostly by virtue of being a high expression gene whose name is first in the alphabet, there's a big accumulation of ribosome footprints right at the annotated start codon and essentially complete depletion over the coding sequence, whereas when we look at the footprints of elongating ribosomes, they're all along the gene. So we took this pattern of footprint accumulation right at and sort of trailing off just after the start codon and turned it into a predictor for start sites. And so we trained it on a number of annotated start codons, and we tested it on a separate set of annotated start codons of different genes, and we found that we could successfully predict about 85% of the annotated start codons with an extremely low false positive rate of predicting start codons elsewhere in the middle of the gene. And since it worked well on these annotated start codons, we sort of turned it loose over all the rest of the data to see where initiation was happening besides the places that we expected it. And you can see in ATF4, for instance, it picks out the 2 upstream AUGs. It actually indicates, as we sort of know, that there's very little initiation on the protein coding gene under these conditions. And it also detects sites of initiation at this sort of upstream non-AUG position that we saw before.
What went into the prediction?
What went into the prediction is just a profile of the Harringtonian footprint counts from 3 different replicated samples, basically. And so nothing, actually none of the data from the elongating ribosome footprints went into that data, and nothing about the sequence went into that prediction, which comes up in a minute, because we can see that even though we did not look at the RNA sequence, there's a massive enrichment for AUGs, about 35-fold, under our sites of initiation versus their overall abundance in the transcriptome, which is very nice to see because translation predominantly initiates at AUG codons. And we also saw an enrichment for a spectrum of other near AUG codons as well, that we could see initiation at CUGs, GUGs, to a lesser extent UUG and ACG. And this is slightly different than in yeast, where it seemed like UUG was the best non-AUG codon. But this sort of set of codons, and we can potentially explain that. I told you that the start codon is recognized by base pairing with the initiator tRNA, and it could be that all of these can favorably mispair with the initiator tRNA in a way that allows initiation to happen there, albeit at lower efficiency much of the time. So this spectrum, we could actually see that it was different if we looked at upstream start sites versus downstream start sites. So I showed you many cases where we saw initiation in the 5' UTR, and these were often occurring at non-AUG codons, and in large part that's because there's pretty strong selection against AUG in many 5' UTRs. Doesn't fully explain this bias though. And when we see initiation in the middle of a gene, that much more often happens on an AUG codon. And so one explanation I think that we would favor for this is that there are different initiation complexes that have different stringencies for start codon recognition. So the idea is that some of these are high stringency, and so they are rarely going to initiate at a non-AUG codon. So they're going to skip all the stuff in the 5' UTR. They're sometimes going to bypass the first AUG they encounter, but because they're high stringency, when they do initiate inside of the gene, they'll almost always initiate at an AUG codon. And in contrast, the low stringency initiation can more often happen at some of these non-AUG codons, but that when it happens, but because it's low stringency, it rarely skips the first actual AUG that it encounters. So we don't have sort of direct evidence for these different complexes, but certainly some process like that could explain this strong difference between what we see in 5' UTRs and what we see inside of genes. So I've talked about this upstream initiation mostly in the context of sort of regulatory translation events that are essentially decoys that block the ribosome from reaching the coding sequence, but there are times when sort of extended forms of a protein, it actually produces a larger form of that protein. And here's an example in the mouse SWI5 gene that this is the annotated coding sequence which is supposed to start at that AUG codon. we see basically no initiation there, and instead almost all the initiation happens at the further upstream CUG codon, and that's consistent as well with what we see in the profile of elongating footprints. The interesting thing, first of all, this protein, this extended form of the protein actually agrees better with the AUG initiated human SWI5 protein, and it's actually been reported in the literature that SWI5 migrates larger than expected on a Western blot, which would be totally consistent with this CUG-initiated N-terminal extension. And so there are some other known cases of proteins that initiated a CUG codon. The Bob Eisenman found some time ago that the major form of the oncogene c-Myc actually initiates at a CUG codon several codons upstream of the first AUG in the transcript. And in that case, it's not clear this is a functional difference between the two forms of the protein you might make, and I think people found it mostly because c-Myc is very intensively studied. But it seems to be a phenomenon that can happen in other cases as well. And so we looked more generally at the kinds of translation products we saw. And in addition to these extensions that I was talking about, as well as these cases of upstream initiation, which sometimes lie entirely in the 5' UTR and sometimes can overlap the start codon. We also saw cases of, as I mentioned, these internal initiation sites that could make either a truncated protein or could result in out-of-frame translation in the middle of the gene. And the internal initiation is something that is particularly useful to have initiation site data for. So this gene exit is a signaling scaffold involved in innate immunity. And when you look at it, it seems like the entire coding sequence is translated, but when we map out sites of initiation, it turns out that there are really two AUG-initiated upstream open reading frames in this transcript, as well as two different AUGs in the gene producing a longer and a shorter form of this protein. The shorter form is missing the first 115 amino acids, and I mentioned that this was a signaling scaffold. And interestingly, our group was sort of studying the molecular biology of this protein, made a very similar truncation to this, and found that it actually acts as a dominant negative because it binds to some but not all of the components in this signaling cascade. And here it looks like this truncation that they made purely synthetically is something that actually happens in the cell normally. And this is something that in the specific case of the transcription factor CBP alpha was actually known to happen. This has been pretty well studied by the Kalkoven group, and there are actually 4 different initiation sites on this, but there are 2 major ones here, B and C. And the B initiation site makes a full-length transcription factor with an N-terminal activation domain and the C-terminal dimerization and DNA binding domain. There's also a downstream start site that produces a form that just has this dimerization and DNA binding domain without the activation domain. And they've done some work to show that a single full-length transcript produces all 4 of these different protein isoforms. And it turns out that the full-length form of this transcription factor is an activator that has an antimitotic effect in liver cells and in adipocytes, whereas the truncated form, again, serves as basically a dominant negative because it can dimerize and bind to the same DNA sites as the full-length protein, but it can't actually activate transcription, and so it has basically a chromitotic effect. And there are actually switches between the upstream and the downstream initiation site depending on growth conditions. And so there are a number of reasons that it might be interesting to regulate expression of a protein actually by producing 2 different isoforms off of the same RNA. First of all, it's something that the cell can change very quickly. Here the cell wanted to switch from the full length to the truncated form. It doesn't need to produce a new RNA. It doesn't even need to wait for the existing full-length protein to turn over. It can just immediately switch to producing an inhibitory form on the same transcript. The other thing that's interesting is that transcription factors are often fairly low abundance proteins, and when you have a low abundance, a lowly expressed gene, the absolute levels of its RNA in the cell are relatively small, and as a result, there's a lot of variation cell to cell in the absolute number. If the typical cell has 2 copies of the CEBP transcript, there are going to be many cells that happen to have 0 or that happen to have 4 at any given time. And if you're actually producing both a protein and its dominant negative from the same transcript, then you're probably buffering that cell against cells against that variation. Where you have more of the RNA, you produce more of both proteins. So.
Do you have any data on how these things are regulated at the protein level? Can you make any generalizations about how the dominant versus the non-dominant starts are regulated?
Yeah, we'd like.
You may not have.
In this particular case, it actually can be affected by acted by signaling through MTOR. That under conditions of elevated MTOR activity, you, I believe, tend to pick the downstream start site. It's not totally clear why that's true, but that's the observation. It probably responds to some other signal. I think we don't know what they are.
And the degradation?
I don't know anything about the degradation. I mean, it might be known. I don't know. And then the last thing that we see is cases of annotated non-coding transcripts that actually do seem to be bound by ribosomes. So this is a case of a annotated non-coding transcript, and here's sort of the genomic view of the RNA that they annotated and the positions of ribosomes and also sites of initiation. And when we go and we actually look at just the transcript specifically, we can see that the 2 major sites of initiation that we map with Harringtonin lie on the first 2 AUG codons in the transcript, that elongating ribosomes show up on essentially the first 2 open reading frames and not really any further into the transcript. And that the first reading frame on here is actually quite short, but the second one is 59 amino acids long and is almost certainly large enough that it could fold into a specific structure and carry out some specific function in the cell. There are other proteins, such as PGC, that are not much bigger than this and definitely function at the level of proteins. So we don't know that in this specific case that it's functioning as a protein, but it's certainly possible.
Why this gene was annotated as a
yeah, I think that basically that this coding sequence is pretty short and presumably is not very well conserved. And those are the 2 things, or I guess, more than that, well conserved, that the amino acid composition might not look typical for a protein. Those are the sorts of things that people tend to use when they're trying to discriminate non-coding and coding transcripts.
Because people look at conservation of the thought position of columns, and
yeah.
There should be less conservation in this position.
Yeah. Yeah, and I for this particular transcript, I don't know what the conservation profile looks like.
So this protein, if you look in genetic, this protein is not, there's no, there is no, this protein does not exist, or it hasn't been seen?
In this particular case, I don't know. There are definitely proteins on, there are transcripts that were initially annotated as LinkRNAs where people have definitively detected the protein product. I mean, again, even then, it doesn't tell you that it's functioning as a protein. It's telling you that it's being translated. And we can see, some other cases too. This is a gene that's interesting, mostly because it has actually a characterized phenotype. And again, we can see, it was actually, some databases call this non-coding. Some say that this larger 81 amino acid reading frame is the protein here. We don't see it being translated at all under these conditions. And it seems to be these 2 further upstream coding sequences that are. Again, 54 amino acids is probably big enough to function. 25 amino acids could be big enough to carry out some function. And so in general, I think what I would say about the presence of ribosomes on things that look like LinkRNAs is it's probably a whole spectrum of different effects, ranging from cases where annotation efforts simply missed a perfectly ordinary protein, through cases where essentially, if the RNA is present in the cytoplasm and is a cap and polyadenylated RNA, it's probably going to be engaged by the ribosome at some level, whether or not it encodes it sort of functions by encoding a protein. And so you can get translation. In some cases, that translation really falls very clearly in open reading frames. In other cases, it's weak and it's in a number of sort of overlapping reading frames. And I think especially in those cases where it looks much messier, those are probably the cases where they aren't functioning as a protein. It could actually be that translation is still functional. For instance, RNAs, sometimes the translation of an RNA can provoke their decay. There are certainly the cases of non-coding transcripts that are translated specifically so they can be turned over. And of course, there are real things, active structural RNAs in the nucleus that are just never bound by ribosomes because they're never even in the same compartment as ribosomes. So it's really, I guess, a whole spectrum of things, and I think that we're going to see a lot of different functions for LinkRNAs emerging as we get a better handle on them, and one of those might be to encode the production of proteins that we had a hard time recognizing as being proteins. So one question that you might worry about, that we certainly worried about, is whether these, especially like these non-AUG start sites, are somehow an artifact of the drug we're using, herringtonin. So it is a drug that affects the ribosome, and you could imagine that it's somehow changing how the ribosome works in addition to freezing it at sites of initiation. And so fortunately, there are actually a couple of other groups who have used different drugs to look at similar possibilities. So the Bosch lab actually used pyrimycin to try to map out sites of initiation. So pyrimycin, in these pictures did not come out well. So pyrimycin actually doesn't affect initiation at all. It poisons elongation, and if you treat with very high doses, and so what it does, it causes ribosomes to terminate early. And if you treat with very high doses of pyrimycin, the ribosomes will terminate very, very early, and really you'll get the same sort of pattern that we see with herringtonin, where there are a lot of ribosomes right at the start codon and it trails off very quickly. And so if you look at where ribosomes remain after you treat with high doses of pyrimycin, you can map out sites of initiation, similar to what we were able to do with herringtonin. And they see actually a pretty similar result, that they see enrichment of AUG codons is what's here in red, CUG codons in orange, as well as some of these same other codons that we saw at GUG, UUG, and ACG here. And they also saw a similar pattern of a substantial amount of UTR translation, is what that says there, as well as canonical initiation sites, and some cases of CDS overlapping stream open reading frames, these overlapping ones. And there's actually also work from Xubing Chin's group using the drug lactamidamycin. So this is, again, it's an initiation inhibitor that acts a completely different mechanism. It actually binds a completely different site on the outside of the ribosome to immobilize the ribosome, again, at sites of initiation specifically, and they show the same sort of pie chart that you can see, strong enrichment for AUG codons, substantial amount of CUG, other codons that are basically one nucleotide off from AUG, and they don't have a pie chart. They, again, also see the spectrum of a substantial amount of initiation in 5' UTR, somewhat less in genes, and some of it actually causing protein isoform diversity.
Is there a way to stop the ribosome anti-site of contamination?
Not that I know of. I've ordered a bunch of odd drugs that are supposed to affect the ribosome and tried them, and none of them seem to. There's a drug that might do that in bacteria, but I don't know of it. And actually, the Sagatellian lab recently characterized some protein products from some non-canonical translation by mass spectrometry. And so here is just their sort of schematic. What they saw, that in yellow here are the peptides that they saw compared to the blue annotated coding sequences. So they could see some peptides derived from 5' UTRs, from out-of-frame translation in the gene, from an annotated non-coding RNA, and from an antisense transcript. and they could attribute these to translation in 5' UTRs, and they could see that many of them are AUG initiated. They saw other initiation codons as well, and in many cases they weren't able to detect actually the site of initiation, specifically by their mass spectrometry data. So I think that this is, yeah.
I guess just I've been trying to think about this. So there's a lot of there are, at least I'm from a East Lab, and I know that there, like from my experience, there's a number of instances in which there's I guess unexplained conservation in non-coding regions. And I'm just wondering, like you keep presenting like evidence that there's transcripts that extend beyond, I guess, what we would like consider the coding region. Like how much possibly some of this conservation in these non-coding regions could be attributed to, I guess,
alternate protein products being.
Right.
Yeah, I think that that's definitely possible. Yeah, I think that. I mean, and there are cases. There are a few cases in yeast where it's actually known that there are sort of extended protein forms made from alternate initiation. Yeah.
Just kind of along the same lines.
Yeah.
Yesterday we had a talk about cancer cells, and eRNAs were mentioned, short RNAs
Yeah.
and transaminases. Do you see any kind of translation happening in cells?
I would no, we don't. I mean, I don't think we've looked really. We haven't looked really systematically for enhancer RNAs. We have looked for essentially like random, orphan regions of the genome that seem to be translated, and we don't see that. I think the only things that end up being really translated are things that are polyadenylated and exported to the cytoplasm. And I think enhancer RNAs are not polyadenylated and exported. So basically anything that we see translated, we also see in polyA selected RNA-seq data. So I mentioned that Harringtonin blocks translation initiation, and one of the things that we wanted to use it for was actually to map out sites of initiation, but the other thing is actually our original motivation for finding the drug is actually to look at rates of elongation in cells. And the idea here is that if you treat with this drug, showing you all the ribosomes sort of running off the transcript, but that you can actually treat with it for a very short time and try to see how far the ribosomes move in that short time. And so I showed you here this profile where we saw dramatic depletion of the ribosomes after we treated with Harringtonin. But if we now look at a couple of, I guess, 3 sort of randomly selected long genes, and we look at the ratio of footprints in Harringtonin to untreated, we can see that as we look out 1,500 or 2,000 codons out in these genes, we actually see that the depletion recovers. Our interpretation is that basically, though we stopped initiation and let elongation run for a few minutes, that these ribosomes actually take many minutes to synthesize these large proteins, and actually these ribosomes just still haven't finished. And so we wanted to actually use this as a time course. You treat with the drug, and then you watch the progressive runoff of ribosomes. You just measure the size of the depleted region as a way of measuring the speed of elongation. And so we were able to do a time course. I think these 30-second time points are about the fastest you can do for just technical reasons and doing the experiments, getting the drug into the cells. And we could measure the extent of depletion and actually fit an elongation rate of about 4.8 codons a second as the rate at which the ribosome depletion runs down the transcript. And this was largely the same for different classes of genes, but it doesn't have really high resolution. Of course, I'm saying that 30 seconds is about the fastest pair of time points that we can look at, and the ribosome is moving at about 5 codons a second, so that's actually 150 codons between each time point. And so there's a lot of room for sort of more local variation in the rates of elongation. But one of the things that this was important for in our thinking about the data is that we and other people always associate the presence of ribosomes on an RNA with the amount of translation going on. That is to say, if you see more ribosomes, you assume it's because there's more protein being made. whereas if you have a lot of ribosomes that are stalled non-productively on that RNA, you'd be fooled into thinking that a lot of protein is being made when nothing was actually happening. The fact that we treat with herringtonin and we see ribosomes run off of practically every RNA that we look at, I think is telling us that there aren't a lot of persistently stalled ribosomes, at least in our cells and in our conditions, that you shut off initiation and everything goes away in the course of a few minutes. And that, at least in sort of bulk measurements, everything elongates at about the same speed. So it isn't the case that when we see twice as many ribosomes on one protein, it's because that protein takes twice as long to synthesize for some reason. So that's sort of the very coarse-grained view of rates of elongation in vivo, but there are cases where we know that the ribosome actually stalls for quite a while during translation. I alluded to some of those earlier. One of them happens at the C-terminus of this protein Sec61B, and so it turns out that this is a transmembrane protein whose transmembrane helix is at the C-terminus, and in order to be properly inserted into the membrane, it needs to stall with that C-terminal helix, like shown here, still stuck inside the ribosome for a while. Yeah?
Just going back to the point you just made, and you're talking about, so let me get this straight. If there are, regardless of the size of the protein and regardless of the number of ribosomes that are present, translation is occurring at approximately the same time, and number of ribosomes and length of protein is
yeah, so the codon.
Well, longer proteins are going to take longer, but it's not.
Yes.
It's not a function of the number of ribosomes.
Right. It doesn't seem to be. And I guess one important point is that even the most heavily ribosome-loaded RNA, the ribosomes are still spaced well apart. So that we don't see a lot of evidence for ribosomes like running into each other or anything. So it seems like each ribosome is acting pretty independently.
And I guess kind of earlier you mentioned something about like if there's like between these like alternative I guess isoforms where there's like a longer version of the protein and a shorter version.
Yeah.
That I mean, is there like I think some people have started doing this. I mean, how do you attribute that to like an actual real fitness cost of a cell? Like, can you do that? And how would you do that?
I think, I guess what I would say, first of all, I think the questions about, really the functional impact of these, we don't have a really good high throughput way to look at that, right? That we basically need to, if we think something's important, I think the profiling data is pointing us at mutations that we could make and test. And that's really, in my mind at least, what it mostly comes down to. We can see these phenomena, but what they're doing for the cell or whether they're noise in some cases, we don't know.
So do you think this 5 photo per second is constant across species?
So people have tried to estimate this in a few other ways, and it's roughly consistent with what people see in animals. I think it's slightly faster in yeast, and it's a lot faster in bacteria. Bacteria is probably 20 codons a second. But they pay a cost for that. Bacterial translation is not nearly as processive as translation in eukaryotes, so they just can't make really big proteins. And it might be in part that going at 5 codons a second is what you have to do to get that extra processivity to make some. And there's this muscle protein called chitin that's like 20,000 codons long, right? So you need to be really processive to make that properly most of the time. Yeah.
So coming back to your ESL experiment, so if you say that the efficiency of translation increases, does that mean that you get more ribosomes on the same mRNA molecule, or just the number is constant, just the processivity increases?
I think it's more ribosomes per RNA molecule, definitely, yeah. So we wanted to look at pausing at specific sites on transcripts. We sort of talked about before cases where we're looking at some profile for a specific gene. You see a big accumulation of ribosomes at one spot versus everywhere else. And we can, in fact, see exactly that sort of accumulation right at the C-terminus, the stop codon of Sec61B. And so in addition to looking here, we can also actually look at a case where translation is known to stall in the middle of a gene. This actually happens on this protein XBP1, which is regulated in this kind of Baroque way. But its RNA needs to be brought to the surface of the ER, and the way that happens is that the XBP protein has a hydrophobic helix that sticks to the surface of the ER. And so while it's still being synthesized, this hydrophobic helix brings the ribosome that's synthesizing it and the RNA template all to the ER surface. And of course, this is more effective if the ribosome stalls during translation for a while. And they were able to show in an in vitro translation system that thing will actually stall for like 20 or 30 minutes. Probably doesn't stall that long in the cell. But what we can see is at exactly the position where they detect this in vitro stall, we see a very large accumulation of ribosomes sitting right on this codon 256 of this gene, which again, we think points to the fact that where people have genetically defined stalling sites in proteins, that we can pick them out, and gives us some confidence that we pick out other sites where we see big accumulations of ribosome footprints. Maybe we can figure out what things tend to make ribosomes stall. So we looked at some of the things that people might expect. Things like codon usage and RNA structure didn't actually have a big impact on these cases of really strong ribosomal stalling. Actually, the thing that had the biggest impact was peptide sequence. And so what we found is that typically when the ribosome has just synthesized 2 prolines in a row, and when the next codon is something like a glutamic acid or aspartic acid, it seems to pause the ribosome quite effectively. And this actually is some work from Alan Buskirk showing that polyproline sequences can pause translation in bacteria. And more recently, it was actually discovered that a somewhat mysterious translation factor called EIF5A is actually absolutely essential for bacteria to be able to translate past polyproline codons. If they don't, polyproline stretches, if they don't have EIFP or the eukaryotic homolog EIF5A, you simply can't make 3 or more prolines in a row. Of course, proline is a very strange amino acid. It has a very odd and constrained configuration. It probably just means that the geometry of the peptide bond formation in the ribosome is poor when you have many prolines in a row. It could be other things about the chemistry or the details of the structure, but it's something that certainly stands out in our data is really sort of the peptide signature that we can associate with pausing translation. So this is really the only thing we were able to pull out at sites of really strong stalls. We were also interested in especially sort of more subtle effects from codon usage on the speed of elongation. So this is something that people think about from a number of perspectives. I mean, so the things we know about codon usage are that there's a lot of selective pressure to use a certain subset of codons, especially in highly expressed genes. It's really pervasive across different organisms. The favored codons differ, but the presence of codon bias is strong. When you go to express heterologous genes at high levels, optimizing the codon usage can decrease toxicity and increase protein production. And the other thing we know is that the favored codons tend to have abundant tRNAs. And so many people, I think, assume the idea is that if you have a lot of a tRNA, you can decode that codon quickly, and that's favorable for producing a lot of a protein. And we wanted to look in sort of some detail at the impact of codons on translation, and so in collaboration with Daphne Kohler, a statistician at Stanford, we went to analyze some of these footprint density profiles in yeast. And we were hoping to get 2 things, to some extent better estimates of gene expression, rather than just averaging across, the number of footprints across a gene. Could we infer a number of ribosomes, but more importantly, to get a better understanding of the speed of decoding and ask about subtle effects that could come from structure and codon usage. So of course the thing we observe in this model is the number of ribosome footprints on a given codon in a given gene. And what we assume is that there's a protein synthesis rate for that gene, a number of ribosomes that are translating it, and there's a dwell time that the ribosome spends at each residue. And then the thing we observe is essentially the amount of time you spend at that residue. It's a number of ribosomes going across the gene times the amount of time each one spends at that codon. So those are the things we wanted to learn. And we essentially assumed that the dwell time was determined by, essentially by the codon in this case. And so that we actually, we observed the occupancy, which comes from, which is a count that we infer from that we observe, and we can infer the occupancy of the residue, and from that we can infer these parameters. And these codon-dependent parameters essentially tell us how long the ribosome spends at that codon on average. And as I mentioned, these common hypotheses that tRNA abundances are limiting for elongation. And we wanted to know if the measures of codon adaptation in particular, this one we're looking at the TAI, which is the genomic tRNA abundance, is correlated to our rates in yeast. And the answer is that there is some correlation. So certain codons that are translated very slow, so on the x-axis is a observed translocation rate, and the y-axis is a tRNA adaptation. We can observe that some codons with low tRNA copy numbers are very slowly translated. Some tRNAs with very high copy numbers are translated more quickly, and it's clearly significant, but it's not, it's not perfectly explanatory, certainly. But because this was done in yeast, we also had actually the ability to manipulate tRNA levels pretty easily. So you can knock out all but one copy of an abundant tRNA and decrease its level quite a bit, or you can dramatically overexpress a tRNA. And so we were able to do both of these things. Here, the x-axis, I'm showing you the elongation rate of the per codon elongation rate. So each point now is a codon, and we're comparing the wild type elongation rates with the elongation rate in a mutant. We're overexpressing the tRNA that codes AGG 20-fold. You can see is that AGG in red is pretty slow normally, and it doesn't change at all when we overexpress it 20-fold. We believe it's being charged properly. We know it's abundant as a mature tRNA in the cell, but it doesn't seem to speed up decoding of AGGs. Likewise, we can knock out 3/4 of the tRNAs that decode the ACA codon, which is normally a pretty fast codon. We actually do perturb decoding rate some by doing this, not dramatically, but most importantly, the ACA codon itself is still one of the fastest decoded codons across genes. So, our conclusions in this, first of all, are just that tRNA abundances are not really the rate-limiting factor for codons for elongation. And there's actually some other evidence by ribosome profiling and more genetic approaches suggesting that one of the major things that determines whether a codon is fast or slow at decoding is whether it pairs perfectly with its tRNA or not. So, of course, there are 61 sense codons that are used in proteins, but not all of those have a tRNA that matches them exactly. In some cases, you'll have a GU pair, wobble base pairing, and in some cases you actually have a modifier like an inosine in the anticodon that can base pair even more broadly. And those wobble base pairings tend to be decoded somewhat more slowly. And this still sort of raises the question of, why is it that we see such a strong selective signature for codon usage, especially highly expressed genes, and why does it correlate with tRNA abundance if tRNA abundance isn't driving the decoding rate? So our conclusion is, the standard model I mentioned is that low tRNA abundance leads to slow decoding. Slow decoding drives codon usage bias, and I think we have some pretty good evidence against this and suggest that perhaps what's actually going on is the other, that slow decoding because of wobble base pairing, because of features of, for instance, the tRNA body. So actually when the tRNA decodes, it actually fits into the ribosome and actually goes through some pretty dramatic conformational changes during elongation. So effects like that might mean that some codons are bad to have, especially in highly expressed genes. And so because these codons are coded slowly and otherwise cause problems during translation, that codon usage bias arises, where you get rid of these bad codons, especially in genes that are highly expressed, and then once you no longer use these, you no longer have pressure to maintain high levels of the tRNAs that recognize those codons. And in fact, you would actually probably have pressure to decrease the abundance of those tRNAs, because a high abundance tRNA that's used infrequently might actually promote misreading during translation. So I think that this is at least consistent with what we see. And hopefully we'll, in the future, be able to get sort of a better handle on how all of these affect. If it's something about tRNA bodies or wobbly coding, again, that is something that we can, in yeast, replace all of these tRNA features and see if they affect decoding and see if they affect the use of, the impact of these codons in high expression genes. And so with that, I'd like to wrap up and thank some of the people in my own lab who've done some of this work, as well as people in the Weissman lab where I developed ribosome profiling and had a number of excellent collaborators. Liana, a bioinformatician at Stanford, a lot of help with the bioinformatics, especially for the mammalian data. Daphne and Christina I mentioned for the model of elongation. Jeremy and Julie for help with ES cells. The microRNA work was principally the work of Felix Guo in Dave Bartel's lab. The UPR was done in collaboration with Carmela and Peter Walter at UCSF, and we also had some help from Gary Schroth and Shujun Lo in developing the technique from Illumina. And so thanks for your attention, for all the excellent questions, and hope we get a chance to talk some more about this stuff.
Yeah, there's some popular rockstar 5' UTRs that people put on genes like globin and bovine growth hormone or whatever.
Yeah.
Does your data say that that's more about the stability or more about really good initiation?
I would say probably some of it is really good initiation. I think that having a short and unstructured 5' UTR with a good initiation context can make a big difference. Certainly one thing that's true is in cases of like these non-AUG codons, having the right sequence context around them is very important. So, I think that the Kozak consensus isn't. I guess what I'd say is that like in terms of sequence context, that you need to not have a bad context around an AUG to get it to be used well, but you need to really have just the right context around a non-AUG codon. And more broadly, in terms of these very efficient 5' UTRs. It's possible that there are other more specific features about them besides the absence of structure and the and being relatively short, but I don't think we have a great handle of it.
This is a little bit outside of something that just certainly not in your data.
Okay.
If you look back at particularly the mammalian data, are there structures in the DNA or functional elements in the DNA that sort of talk to what goes into a UTR that's more likely to be, or into a protein that's more likely to be initiated at multiple sites?
Not that I know of. I guess the one, and there's one interesting case of a promoter element where essentially like the sequence, sort of an odd case, but essentially the promoter sequence element is also a translation enhancement element when it sits right at the very beginning of the 5' UTR is, comes from the promoter, and so this element actually does both. But more generally, not that I know.
Questions?
Yeah.
One less practical question, or one more practical question. Is this something that we're going to be able to see when we just sort of analyzing regular mRNA
So in some cases, in all the cases I talked about today, we looked at transcript structures and everything, and we really believe that it's alternate initiation on a single mRNA isoform. But I mean, one big thing that causes different protein isoform, different first exons can lead to different protein products that have different start sites, certainly. And so that, I mean, you definitely pick up an RNA-seq data, and there's probably one significant thing driving some of the alternate initiation. Yeah.
You mentioned something about Fourier, mRNAs that have Fourier tail, of the ones that are translated, the ones that don't have Fourier tail, not translated.
Yeah.
Do you see an effect of alternative Fourier dilation on translation?
This is maybe one of the real limitations of ribosome profiling, is that you can't tell what the 3' UTR of that message was. So if you have different poly-A sites in the same cell, you get just the footprint itself, and it doesn't include poly-A tail, so you can't tell how it was translated. I think you need to have conditions where you knew the polyadenylation site changed and you saw a change in translation, for instance. You can't directly infer that. Yeah.
Might be a completely nonsense question, but do you actually see non-coding genes that are ribosomes, so that are annotated as non-coding?
We do see some. Yeah. So some of the cases I was talking about, these short reading frames, and those are on RNAs that are annotated as not coding initially. So I would say is that so there definitely are cases like that, and I think they fall into some of them. It probably really is that there is a specific short peptide being translated off of that non-coding RNA, and others I think it's more because that RNA is present in the cytoplasm. There's some amount of translation around the 5' end, and it sort of looks weak and messy translation like we see in any 5' UTR without a major translation.